Hello, I'm Dr. Charles Knight, and this is another installment in my 10 lectures on evolution. I'm recording these for my Bio 414 evolution class. I work at Cal Poly in San Luis Obispo, California, and I'm in the Bailey College of Science and Mathematics in the Department of Biological Sciences. So the paper I'm going to present today, this is our target paper number 5 in the course. It was published in Science on October 21st in 2016. For this class, this is a fairly old paper now, but the title is Predictable Convergence in Hemoglobin Function Has Unpredictable Molecular Underpinnings. It was by these authors. I've been using this paper for a number of years in my evolution class because I find it fascinating whether or not there is a predictable molecular basis for convergent phenotypic traits. In this case, we're going to be looking at hemoglobin function as the convergent trait. And the answer is yes and no, really. So that's just kind of a foreshadowing. But as I do, I'd like to present a few of the authors of this study. Was led, I think, largely by Jay Stortz at the University of Nebraska, Lincoln, where he works with Chandrasekhar Natarajan, who's also a principal investigator at the University of Nebraska. They collaborated with Christopher Witt at the University of New Mexico to complete this research, which I think is a great addition to molecular evolution and the basis of convergent evolution, the molecular basis of that. And I really appreciate the work that all of the scientists did in producing this short paper in Science that's very ambitious in the sense that it covers both comparative genomics, really some detailed protein physiology, and then even some re-engineering of ancient proteins. So it's a really cool study. Thanks a lot for producing this. In addition, for the preparation of this presentation, a commentary or perspectives article by Jamie Bridgeham that was in the same issue of Science was also very useful. It kind of helped explain the story to a larger group of people. It helped me think about what was important for this presentation for my students too, and I appreciate that. Thanks for that. And also, I'd just like to mention at the beginning that I'll have some textbook tie-ins to the textbook that we use called Evolutionary Analysis by John Heron and Scott Freeman. And of course, at times I'll have some information, background information from Wikipedia and Google that I'll layer in too. So I always like to start with some take-home points of things that I learned having gotten into this paper and really examined all of the data and the figures. What I found that I think is important to take home for my students is that high elevation birds have convergently evolved greater oxygen binding affinity of their hemoglobins, and that's both HbA and HbD. We'll get into the weeds here on the details of these two different hemoglobins. But high elevation birds in general have greater oxygen binding affinity. That's convergent. It was very well shown in this paper. These decreases, and we're actually going to be talking about these increase in oxygen binding affinity as a decrease in P50. So these decreases in HbA and HbD P50 were only rarely due to homoplasy, which is parallel molecular changes. So that would be a predictable molecular underpinning if there were a predictable parallel molecular change that caused that decreased P50, but in general that wasn't the case. It happened a few times, but not in general. Then they were able to re-engineer ancestral proteins, these ancestral hemoglobins, and confirm that a few key amino acid positions can result in decreased P50. Notice those two qualifiers, few can, not a general story, but a few can. But these mutations were contingent on the ancestral background genome, and that allostery really mattered, how the mutations were affecting the active site from a long distance away. That's allostery. So There's obviously some details here that are going to have to be unpacked for the general reader to understand, and we will do that in this presentation. So I always like to start by reading the abstract just so that we start at the same basic beginning. Um, the authors put a lot of effort and thought into what to put into this abstract, and it's actually very readable. And then I'll explain some of the complicated terms and phrases in the abstract. So I'm just going to read it slowly at first here. I'll read the title again. Predictable convergence in hemoglobin function has unpredictable molecular underpinnings. To investigate the predictability of genetic adaptation, we examined the molecular basis of convergence in hemoglobin function in comparisons involving 56 avian taxa that have contrasting altitudinal range limits. Convergent increases in hemoglobin oxygen affinity were pervasive among high-altitude taxa, but few such changes were attributable to parallel amino acid substitutions at key residues. Thus, predictable changes in biochemical phenotype do not have a predictable molecular basis. Experiments involving resurrected ancestral proteins revealed that historical substitutions have context-dependent effects, indicating that possible adaptive solutions are contingent on prior history. Mutations that produce an adaptive change in one species may represent precluded possibilities in other species because of differences in genetic background. These are some of the phrases that I think are important here. Parallel amino acid substitutions, key residues, resurrected ancestral proteins, context-dependent effects, and contingency in general, and precluded possibilities. I'm going to have a glossary slide next that kind of helps with this. All right, so these are my glossary notes for the abstract. Parallel amino acid changes are convergent amino acid changes that are not due to common ancestry. I want to introduce the word homoplasy, which is a similarity that's not due to common ancestry. It's the opposite of homology. Homology is a similarity that's due to common ancestry. Homoplasy is the opposite of that, any similarity that's not due to common ancestry. I know I'm harping on that point. It's important. Key residues is a way of referring to amino acid positions under strong selection and that are functionally important or constrained. Those would be key residues. It was kind of older protein biochemistry when they called them residues. Now we mostly call them amino acid positions. Same idea though. Resurrected ancestral proteins are proteins they made from inferred ancestral sequences, sequence information alone, and then they reconstructed those proteins, which is a pretty impressive biochemical trick that they were able to do. And lastly, contingent or contingency is a word and phrase I'm going to explain more on the next slide, but basically that history matters, or that mutational paths are context dependent. The contingency of mutations depends on the genomic background. I want to go to Stephen Jay Gould, and my introduction really to evolutionary biology came from Stephen Jay Gould in this book, Wonderful Life, in 1989, where he really introduces this idea of historical contingency. And I just want to mention that Stephen Jay Gould fairly recently died in 2002. But he was a popularizer of evolutionary biology for my generation. And he was so famous that he became a character in The Simpsons in 1997. So that just gives you an estimate of his scale of reach in the scientific community. And his legacy really will last with these phrases like historical contingency. He, in this book, suggests that if we replay the tape of time through the Cambrian or through any geologic era, it would be very unlikely to come out the same way again. So replay the tape of time and things are going to be different, or most likely would be quite different than they are today. And this paper suggests that mutations are contingent on the genomic background, like a mutation that seems to be important for the biochemical phenotype of better binding of oxygen does so, but only in certain genetic backgrounds. So allostery, other mutations, are affecting that active site and leading to the increased oxygen binding affinity. I'm going to do a textbook tie-in here now and just some background on hemoglobin in general. We're taking a look here at some red blood cells in the middle, and there's a one that's cut in half on the upper right-hand part of this slide. And I think it's important to realize that hemoglobin is the dominant protein on the surface of red blood cells. There's 27 million hemoglobin molecules on each red blood cell. They're integral proteins, and they're coating this red blood cell. We're looking at human red blood cells, which are enucleated. They don't have a nucleus on the, inside them anymore. They've been enucleated. The nucleus has degraded. That's unique to the vertebrate, well, the human lineage, I would say. I guess primates. I don't know what point in evolutionary history that happened, but we do know that birds are their red blood cells do have the nucleus, so they'd be more spherical most likely. These our red blood cells tend to be a little bit concave, and that may help with oxygen binding ability. Maybe. I don't think it really increases the surface area, but it is, well, I don't know why they're enucleated in humans, but it seems to be adaptive. We can only infer that. In reptiles and birds, they're nucleated, the red blood cells, but that in the end doesn't matter much for this story. Let's take a closer look at the hemoglobin itself, because that's really what this paper is about. This is a picture or a drawing of hemoglobin in the left-hand corner here. Hemoglobin has four subunits of two different types, the alpha and beta chains. Iron is at the center of a heme group, which is coordinated into each of the globin molecules. So I'll just go slow here. It's a tetramer of two different types, we could say, the beta and the alpha chains. So there's two beta chains and two alpha chains. And the heme group is where the oxygen is carried. The oxygen is bound to the iron in that heme group. The heme group is shown in the upper right-hand corner of this slide. And it's a carbohydrate. I described it as it's definitely a hydrocarbon. It's a hydrocarbon. Lots of carbon, some rings with nitrogen. Those are called porphyrin rings. There's 4 of those porphyrin rings that are coordinated and covalently bound with the iron molecule in the center. So this is a hydrocarbon, not necessarily a carbohydrate, but it's a molecule that is coordinated into each one of these subunits of the hemoglobin molecules. Notice that there's 4 heme groups. The binding sites of the heme group to the globin, the alpha or beta globin, may be important active site modifications that may lead to differences in oxygen binding affinity. That's one hypothesis going forward, and actually it turns out that that didn't turn out to be the case in this paper as much as you might think. But I just want you to be very familiar with the hemoglobin molecule, because essentially everything else in this paper deals with this kind of molecule, hemoglobin. 4 different globins of 2 different types. These 2 different types of globins have been produced by gene duplication. They're paralogs, they're homologs that are paralogs, that is, copies within the same species. Let me just read one sentence from the beginning of the paper. A fundamental question in evolutionary genetics concerns the extent to which adaptive convergence in phenotype is caused by convergent or parallel changes at the molecular sequence level. And it is a fascinating question, and it's what attracted me to this paper, and it's the reason why I'm relating this story to you. If it is the case that there's parallel changes at the molecular level that predict the phenotype, that has implications for just the predictability of evolution in general. Um, is it true that specific amino acid changes will lead to a certain phenotype? I think you're probably with me to think that that's a pretty naive concept because each amino acid change has such a small effect, but sometimes there are residues or amino acid positions which might have large effect, and it may lead to these different biochemical phenotypes. And the conclusion of this paper is that it happens sometimes like that, but not all the time. Pruitt in the 1960s won a Nobel Prize for his work on hemoglobin. He really thought that changes at a few key positions would be important, and he kind of predicted the null hypothesis of this paper, which didn't end up to be entirely true. So this paper has really three main parts: a comparative genomics part, a physiology part, and a protein engineering part. And I'm going to use these icons throughout this presentation to let you know what part of the paper we're on. We'll start with comparative genomics. This is a lot. This figure is the first figure in the paper, and actually for my students this paper is unique. This presentation will be unique because I'm going to talk about each one of the figures. Usually I don't have enough time or it's too complicated to present all of the figures, but this time we're going to be able to go through each one. As I normally do, I'm going to break down this figure piece by piece so that we can better understand it. We'll start with the top axis. And the first thing that you should see is there's the two different types of hemoglobin, HbA and HbD, which will be pervasive through this entire story today. HbA is composed of an alpha A globin and a beta A globin. Those two together form a tetramer that form alpha or hemoglobin A. Hemoglobin D, HbD, is a tetramer of beta A globin and alpha D globin. So HbA and HbD share the same beta globin gene, but have different alpha globin genes, or different alpha globin genes. They produce different globins, the alpha A and the alpha D. It's difficult because HbA is a tetramer, but the two types are shown there, alpha A and beta A globin. There's two copies of each of those that produce the tetramer, and then for HbD, there's two copies of beta A and alpha D globin. That's just the basics here. There's a number of numbers that are below each one of those globins, and these are polymorphic sites. Within the species set that they studied, these sites were varying. If there's not a number, for example, let's say 7 or 10, that site was not polymorphic and it didn't carry information, so they just omitted it from the data set just for visual streamlining. So bird hemoglobins come in two different types, HbA and HbD, named after the version of alpha globin paired with the beta A globin. That's alpha A or alpha D. The beta globin is always the same, beta A globin. There are only the polymorphic sites will be shown for each species. Each globin is about 140 amino acids long. It's interesting to note that each one of those globins is about 140 amino acids long. And the globins are a gene family of paralogs produced by gene duplication, but function together in various quaternary structures. Now we move on to the left-hand axis, where we see that there's 29 species pairs with a high elevation species and a low elevation species or counterpart. The high elevation species is shaded blue. The one that's shaded white is the low elevation counterpart. And in some cases, the pair was conspecific. So the same species at high elevation and low elevation, they haven't diverged enough to call them different species, but those are shown here in red, where we have conspecific pairs, where one high elevation population is compared to a lower elevation population. This is what we call a species pairs approach, and it's an incredibly useful technique for studying adaptation because it's so much more robust than just studying one high elevation species compared to another lower elevation species, where whatever you saw in the high elevation species could be just a fluke or a chance event. How do you know that it's a replicated divergence? This kind of methodology of looking at 29 species pairs is an incredibly robust way to get at convergent phenotypic evolution in high elevation. So kudos for doing that. So now we see the first molecular data. These are amino acid uh, identities at each one of these loci, which is the protein position. Um, and remember, only polymorphic sites are shown for across this entire data set. In some cases, for the first pair that I'm revealing here, you can't even see the names there. It's covered up by the comparative genomics icon, but the species don't matter that much. But what does matter is if there's a red letter, that's the derived or non-ancestral amino acid at that position. If it's black, that's the ancestral or predicted amino acid at that position. If it's a blank box for both species, that means it's just ancestral. It would be the black letter, but it's just kind of visual clutter to include all of those letters. So they just omitted those ones for the pairs where there wasn't a polymorphism. So what we see here is that HBA has 6 mutations, 5 of which were derived in the high elevation taxa, and 1 was derived in the low elevation taxa. Interestingly, if we look at HBD, there's been 3 mutations, and those were all 3 of them were in the beta alpha A globin. Those were shared with the HBA mutations, but there were no mutations in the alpha D globin. So it's interesting to contemplate then. What does that tell you about the selection pressure on the alpha D globin or HBD? Perhaps that the selection pressure is higher for negative selection so that there's no variation in that sequence in the alpha D globin. Whereas in the beta A globin or the alpha A globin, there is some positive selection, some change that may be leading to a derived hemoglobin in the higher elevation species. Or another way to interpret this is that the alpha A globin is where there's a little bit of fine tuning of the globin that can take place to perhaps lead to greater oxygen binding affinity. I'm revealing more information for more taxa now, and there's take a second to look at the chart now. Look for the red letters, look for the black letters, um, look for other visual elements. I didn't mention on the far right-hand side, there's some phylopic images of the characteristic taxa in those groups. I should say that the phylogeny on the left-hand side is the phylogeny of the 29 species pairs, but it's a broad phylogeny of birds, and it's pretty unbiased. Takes whatever taxa have a high elevation counterpart and a low elevation counterpart, and it's very broad. Another element, if you're looking closely, is that some of the amino acid pairs are highlighted with a box, a black box. And these black boxes indicate where there's been parallel amino acid changes that led to increased oxygen binding affinity of those hemoglobins. You may see some instances where there is also a parallel change, but that parallel change did not necessarily lead to increased oxygen binding affinity. Also, only the ones in the beta A globin are highlighted with a box, but these are where parallel changes underlie an increased oxygen binding affinity of the hemoglobin. I'm revealing the entire data set now, and you can look through it and find more black boxes where there has been parallel amino acid changes. one conclusion that you should make is that this parallelism is not pervasive across the entire data set. Um, in general, there's a lot of mutations that are apomorphic, that is, unique mutations that don't recur over and over again. But as I presented this to my class, they pointed out several instances where there are other places where there's parallelism that I didn't see before, such as the A to T, and it's hard to read, like position 75 or 6 in alpha A globin. But those changes didn't necessarily lead to greater oxygen binding affinity, and so they were not squared off or boxed in the black box. But those residues, especially the ones up at position 83 and the beta A globin, are going to become important later in this talk. So only 4 sites showed parallel amino acid changes that led to increased oxygen binding affinity. So we're going to start asking how those changes led to the differences in oxygen binding. These sites may be near the heme group binding site, but not precisely. We're seeing this is a model of hemoglobin that has the heme group, it's in green, the heme group is in green, and the iron is in the orange circle. And you see the residues where the heme group is binding, and this is not even in the bird hemoglobin. I'm just showing this as an example where, for example, position 63 and 92 are important for the heme group binding. So the hypothesis is that maybe these places where we see parallel amino acid changes are near those heme group binding sites. And actually in the paper, they suggest that these sites that are highlighted in red at the top here now, that is the boxes on their chart, are not necessarily right near the heme group binding sites. So there were lots of apomorphic allosteric mutations. That's a long phrase. Apomorphic means a derived mutation that's unique to a certain species. That's apomorphic instead of synapomorphic, which would be a shared derived character state. And apomorphy is unique to one taxa. So there were lots of apomorphies that involve probably allosteric mutations. Those are mutations that have effects maybe distant from the active site. But those allosteric mutations are still associated with the high elevation habitat. One useful exercise is to go through this table and count the number of mutations that are red for the high elevation taxa. And it's true, looking across the entire data set, there are more mutations that are red, that is derived for the high elevation taxa, but there's still some that are derived for the low elevation taxa too. We don't have much idea of the directionality of evolution, but you could imagine a bird moving from high elevation to low elevation might require mutations that allow it to live at low elevation, just like moving from low elevation to high elevation may require mutations to increased oxygen binding ability. So HbA and HbD together have about HbA by itself and HbD by itself has about 280 amino acids for one pair. Together, well, it'd be more like 560 for a full tetramer. HbA and HbD had mutations in only about 49 and 55 of those amino acid positions. Looking across this table, the ones that are shown for HbA, we have about 49 positions that were polymorphic, and HbD, we had about 55 positions that were polymorphic. And so that implies that many positions are under strong negative selection. There's no mutations that are tolerated within this hemoglobin or that have been produced. There's been a lot of conservation in hemoglobin in general. What I'm trying to get at is that there's a lot of allosteric mutations that may have contributed to decreased or increased oxygen binding affinity. So if we go back to the title, predictable convergence in hemoglobin function has unpredictable molecular underpinnings. The comparative genomic data support this conclusion. Rarely were mutations in these same amino acid positions, but allosteric interactions with mutations at key residues probably matter a lot. And we don't really know for sure what are the key residues. How do you identify a key residue? If we go back to this previous slide, whoop, I don't know, it doesn't let me go back. Those positions that were invariant, that didn't vary, those are probably really key because they didn't change at all. So it's not the case that the places that are polymorphic are necessarily the most important for this evolutionary story, but they are places where there's been changes, and those changes that are correlated with high elevation living may be important, and that's where we're going to go next. I have a few more glossary notes. The heme is an iron-containing carbohydrate, or hydrocarbon, I should probably say, with 4 porphyrin rings, which are nitrogen-containing rings that are bound to beta globin. The allosteric sites is a binding site on a protein that's distinct from the active or heme group binding site. An apomorphy or apomorphic means it's derived but not shared. Homoplasies, any molecular or morphological similarity that's not due to common ancestry. It's very common in DNA sequences since there's only 4 possibilities. For example, if for DNA, there's only 3 possible changes that could be made from whatever base pair it is now. If there's a mutation, it's going to only be one of 3 other base pairs. So homoplasia is actually very common in DNA databases. So now we're going to move on to the physiology part of this paper. They studied oxygen bonding affinity of isolated HbA and HbD hemoglobins from these species pairs. This is their figure 2, which is pretty complicated to see all at once, but the only thing I want you to know from this slide is look at the very top, HbA, the conjunction of alpha A globin and beta A globin. That's what we're going to look at on the left-hand chart, and HbD is shown in the right-hand chart. Remember, it's a different tetramer. We'll start with the left-hand side. This is data for P50 for HbA, and P50 is on both axes, and I'm just going to explain the x-axis first. That's P50 for the low altitude taxon, compared to the P50 for the high altitude taxon. P50 is approximately equal to Km, and I'm pointing that out because in a previous paper we studied Km, which is the Michaelis-Menten kinetic. P50 is similar. It's the oxygen concentration at which 50% of the hemoglobin oxygen binding sites are occupied. So a lower P50 is greater oxygen binding affinity. So again, low P50 is a greater oxygen binding affinity. Now that we understand the axes, and there's one little piece of information, the KCl and IHP were chemicals that they introduced to this blood to make it more like the in vivo conditions. And P50 was measured in torr, which is pressure of the partial pressure of the oxygen. I'm taking a deep breath because it takes a second here. We've got the species pairs and the conspecific pairs, which are the open circles. But in general, each dot is a pair, a low altitude compared to a high altitude bird. When the points fall below the 1-to-1 line, that indicates that the high elevation bird had a greater oxygen binding affinity. Greater oxygen binding affinity because it was lower. It was lower than the low altitude taxon. So all of the points that I have circled in red here have a greater oxygen binding affinity for the high altitude taxa. That's a pretty impressive result already. Just continuing to try to orient you to this chart, that point that I'm highlighting here has the same P50 for low and high altitude taxa. So it's not different at all. But we see in general the points fall below the line, like this one that has a much lower P50 for the high altitude taxa. indicating that oxygen binds much better to the hemoglobin for the high altitude taxa compared to the low altitude taxa. And remember, this is just for HbA. Again, for this pair, the high altitude taxa had a much lower P50 and therefore higher oxygen binding affinity than the low elevation taxa. So that was HbA. Now we're going to move on to HbD. I'll just let you look at this. Think about how they're different. What you should see for HbD is that the points are shifted to the lower left. Immediately, that means that the hemoglobin, HbD hemoglobins, are better at binding oxygen. They're just better. They have lower P50s across the board for both the high elevation and low elevation taxa. However, the high elevation taxa still had lower P50s in general. The points are below the diagonal line. High elevation birds had a better P50 for their HbDs and HbAs. So one conclusion you could make is that why don't high elevation birds just have more HbD in general? It turns out, and they mentioned this in the paper, that from their blood chemistry work, they don't. They have about the same proportion of HbA and HbD. So that's not the solution that birds are using to survive at high elevation. Instead, it seems that, at least from this paper, they're evolving lower P50s of the hemoglobins that they have. Now we'll get into the protein engineering part, which is really, really cool. But to do this, we have to consider the phylogeny of some species and the ancestral species of those species. So what's shown here is a phylogeny of some birds, and there's dotted lines and I just circled in red, ancestral species. And this is a phylogeny of birds, but then the ancestral species are the ones where they reconstructed the hemoglobin of these ancestral species and then introduced specific mutations to see if those mutations led to decreased P50s. Remember this residue 83. That's the focus of the initial part of their protein engineering. I'll go back to the big data set. Remember, position 83 was the one where there was a number of parallel amino acid changes. In general, it was going from a G to an S, a glutamine to a serine, but then on the bottom you see it goes from an N to an S, a different starting point. All right, so this is the beginning of their protein engineering experiment where they introduce this serine mutation to the ancestral genome of the ANC1, the one at the ancestor 1 at the top of the phylogeny. We'll see that again soon. Point here is that in this re-engineered protein, the P50 was about 20. Well, no, it was about 17 or so for the glycine at that position. When they introduced the serine, that reduced the P50 to about 12. And they've got error bars there, assuming a pretty significant reduction in the P50 because of that genetic engineering. So they engineered this G83 to S mutation and produced a significant reduction in P50 in this ancestral background. They tried another mutation on position 867, which was an alanine to an valine, and they show that in addition, that alanine and serine, the double mutant, had the lowest P50. If you look all the way to the right-hand side, the double mutant had by far the lowest P50 compared to any of the other combinations. So they engineered this double mutant and it produced. They tried another mutation on position 867, which was an alanine to an valine. This chart is kind of the take-home chart. If we can understand this, we'll understand the significance of their whole paper. This is the effect of the G83 to S mutation in 5 different ancestral backgrounds. Remember those ancestral backgrounds that I circled in red on the phylogeny. They're on the left-hand side. They're now in the graph, they're shown vertically here. And the axis of the graph is a change in P50. If it's negative, it significantly declined the P50 or decreased the P50. So what we see is in ANC1, that's ancestor 1, which is the species pair near the top. Well, I'm going to start with the ancestral flower pincher. I'm showing where that goes. There was about a 27 degree or 27 a decrease of about 27%, let's say, in that ancestral genome. When we go to the ancestral hummingbird, there was about a 27% decrease in the ancestor 1. The most recent derivation, there was about a 39% decrease. So in the more recent divergences or the more recent ancestral genomes, we see a pretty dramatic decline in P50 with that single mutation, the G83 to S. But if we go further back and introduce that mutation in the more ancestral genomes, we see there's not as much of an effect. So what the conclusion here is that the G83 to S mutation produced significant reductions in P50, but only in some genetic backgrounds, and in general, they were more in more recent genetic backgrounds. So if we go to a conclusion here, we can say that there were lots of mutations at key residues, or lots of mutations not at key residues, and the genomic background mattered, indicating that allosteric interactions are important. Single sites like position 83 and position 67 can lead to decreased P50, but the effect of those mutations kind of goes away through backwards looking time. That is, that mutation doesn't have the same effect in different genetic backgrounds. So were those mutations at key residues for heme group binding? Not necessarily from the conclusion of this paper. Instead, their effects were more allosteric. What is a key residue? It could be those positions that didn't change at all, but these are points where mutations are tolerated within this hemoglobin protein. A few more glossary notes just to wrap this up is P50. Again, that's when 50% of the hemoglobin binding sites were occupied with oxygen. Additive genetic variation is when mutations work independently but together to reduce P50. So each one independently reduces P50, but then if you add them together, that's even more. They work together. Allostery is when protein interactions that are not at the active site or a mutation that's not at an active site has some phenotypic effect on the protein. Homoplasia in general, and homoplasia again is any molecular or morphological similarity that's not due to common ancestry. It's common in DNA sequences since there are only 4 possible base pairs. Again, a mutation of a DNA base pair only has 3 possible base pairs that it could mutate into, so it's likely that it might mutate to the same sequence in another taxa. That's homoplasia. I'll go back to my take-home points. Remember the title, Predictable Convergence in Hemoglobin Function has unpredictable molecular underpinnings. First one is high elevation birds have convergently evolved greater oxygen binding affinity of their hemoglobins, and that's true for both HbA and HbD. These decreases in HbA and HbD P50 were only rarely due to homoplasia or parallel molecular changes. They use protein engineering to show that a few key amino acid positions can result in decreased P50. A few can, but it's not pervasive. These mutations were contingent on the ancestral background genome, and allostery seems to matter a lot. That is, mutations that are not at the active site of hemoglobin can have significant effect, perhaps. More protein biochemistry is necessary, I think, to understand what the key changes really are. And remember, for hemoglobin, it's interesting because the active site is actually on another molecule that's bound into those globins, and it's not necessarily an amino acid, except the amino acids that are binding the heme group. You would have thought that those would be important, but they seem to be invariant. That is, binding of the heme group seems to be pretty solid. There's no polymorphisms, at least from what I understood from this paper, at those particular sites. But there are mutations like at position 83 that are close to those binding sites, maybe, that are affecting oxygen binding in some way or another, maybe changing the pocket of where the oxygen can be bound.